Okay, this is week number 8 in the winter quarter. Only 2.5 weeks left. And today we have the adjunct professor of bioengineering, Professor Tom Daniel, who is a common professor and chair of the newly established Department of Biology, combining 3 programs: zoology, the botany, and biology teaching program. And with more than 50 faculty members, so it's a mega department on campus, with the second largest undergraduate enrollment of 900. So he has a huge job now. But today he will talk about his research. And Dr. Daniel received his PhD from Duke in biology in 1982 with an emphasis of biofluid dynamics. And then he spent 2 years with a postdoctoral fellowship in engineering at Caltech. And then in 1984, he came to UW as the faculty member in zoology department. And now he's a chair of biology. And his research focuses on the neural and mechanical mechanisms that regulate animal movement. And he is particularly interested in the dynamics and control of flight in insects, which he will give us a talk today with a title of Mechanics of Molecular Motors, Modeling Movement, Mostly in Moth.
It's a tongue twister.
Yes.
Thank you, young man. It's a real pleasure to talk before you. And so I have several introductions, actually a plethora of introductions that I'll give you. And the first is that this talk is a little like a marriage. It's a wedding of several disciplines, some biology, some mathematics, some computing, some engineering. We're making a fairly large marriage. And like a wedding, I'm going to talk about something old, which is some early work I did in the late 90s. It seems odd to say that that's old, but it is now. Something new, which is an extension of that work to three-dimensional modeling. Something borrowed, which are the data from much of this talk, and something blue, which is how you might feel at the end. So here's an outline of my talk today. And that is, I want to begin by, why study motion in biology? What's interesting about movement per se, okay? And then I'm going to give a very brief review of an overall scheme of the work we're doing in my lab. And then I want to focus it onto one very specific part of our concerns, and that is, how is muscle designed to provide effective movement and movement control? These two are not separable as we had been treating them in the past. I will talk about some past work on muscle and its molecular motors, mostly from a mathematical standpoint. That is, what sorts of models have come about? What are their strengths? What are their weaknesses? What can we do to address some of these? And then I want to talk to you about new computational work on these problems, okay? So quickly moving on, why study motion in biology? To us, at least in biology, it's a problem that spans all levels of organization, from molecular motors and the kinetics of chemomechanical transduction to how these molecular motors are arranged spatially and how they may interact with one another, to how a muscle may behave in response to a neural stimulus, to how a system of muscles may regulate motion, such as walking, talking, flying, any particular aspect, to the recognition that you cannot understand a neural control of a neuromuscular system unless you understand both sides of that. So, and we'll go on to say it does provide clues to how neuromuscular systems work. And many pathologies require understanding both the neural and mechanical basis of motion. So, for example, if you were a bioengineer interested in developing technology to help paralysis, we have a project that works on paralyzed vocal cords or paralysis in locomotion. If you blindly go in and look at the neural circuitry associated with leg movement, you will probably get the controls wrong, because the neural drivers are running a nonlinear forcing function called muscle. And so how that moves an inertial appendage will have a profound impact to how you will determine what circuits you need to control that. from a biological standpoint, there is nothing more exciting than looking at the diversity of motion we see in biology. And that's not just the diversity of movement from walking, running, swimming, flying, but diversity among kingdoms, bacterial locomotion, and the use of cilia and flagella, both in bacteria and in our respiratory circuit. Plant locomotion and plant movement is in fact a rather unexplored area. Cell division, cell crawling to whole swarms of creatures flying together with their the aerodynamic interactions occurring between individuals. So it's a huge and diverse set of topics. And finally, of course, there's nothing more beautiful than looking at movement in biology, and I say that with no bias whatsoever. From a mathematical standpoint, I have a little bit of cross-training, and that is that in fact, there are some real challenges ahead of us in both computational and mathematical aspects of motion. That is, the mathematics that might describe the fluid dynamics of running or swimming or flying, or the inertial mechanics of a nonlinear oscillator, running a leg or an arm or a jaw and vocal cords. Those are, and I apologize for the informal writing here, but those are cool dynamical systems for which we need to develop mathematical tools, right? And they're complex. However, on the flip side, sometimes very, very simple mathematics can go a very long way. So just how you describe the movement of a human walking in a room might be as simple as a trivial pendulum model to get 90% of the problem done, okay? The remaining 10% are highly complex nonlinear dynamical equations, which we might not want to deal with. And finally, sometimes mathematical and computational tools are about the only tools we have available to study either very tiny, tiny problems, to put them in appropriate context physically, or problems that no longer exist. So if you are Steven Spielberg and you want to figure out the mechanics of a running dinosaur, sometimes your only tools are mathematical models. And finally, from a consumer's view, why study movement? I'll maintain that there are no better robots, so to speak, than the natural ones we see. So to develop a small robotic device, about this large, capable of hovering in my room, in this room here, of coming up to some target, maybe feeding on a flower, I apologize, and hovering there, and then traveling at 35 miles an hour, and then stopping abruptly, doing an abrupt yaw turn, and then chasing a mate and avoiding a predator. I challenge anyone in this room to make a robot capable of that, okay? And I challenge anyone in this room to understand the complete flow of sensory data such robots need to control their movement. So new technologies arise from the inspiration of moving biological systems, new materials, new theories, new structures, and in fact, new amalga, as you may hear later in this talk, of partly biological, partly electrical robotic devices. Okay, so implanting control circuits into a neural system of a moth, for example, might be an interesting biorobot. Okay, so this is an outline of sort of what our lab is worried about, and I'm going to just focus on one aspect for this talk today. There are two choices I can make, and that's give you a little piece of candy on each bit of what the lab does, or just delve seriously into it. So I'm going to delve seriously into one segment of what we're doing, okay? But the group as a whole is deeply interested in movement and movement control, and the two are not separable. So let me show you sort of the flow of our thinking. You have a creature flying in space, and it's receiving massive sensory data. In the case of insect flight, bird flight, or human locomotion, there's a massive amount of visual sensory information that's processed in specific regions of the brain. And in fact, we can find wires going from the visual centers of an insect brain straight to the motor centers. So we can find the connections, we can characterize these, we can quantify how neural systems are encoding visual information, and that affects a pattern of activity from in the central nervous system that drives a system of motor programs. Motor programs running a complex suite of muscles for regulating flight or other modes of locomotion, in our case it's flight, which drive deformations of a skeletal system, which in turn have important mechanical characteristics. And those, which is what the question we're going to focus on in this talk, require an understanding of the mechanism of force generation. Once I am armed with an understanding of force generation and its regulation and its timing, I can then proceed to ask how motions drive structures, propulsors such as wings, and those in turn generate specific aerodynamic forces which propel the animal, moving its position, altering the sensory input, and affecting this systems problem here. Now, I've shown it just dominantly structured by visual sensory information. There's a whole host of sensory data that are flowing on board, but this is just one example. So we have people in the lab looking at visual sensory transduction, motor output, muscle mechanics, the skeletal mechanics, wing aerodynamics, and actually measuring flight forces on insects tethered into little flight simulators. So when you go and play in a flight simulator, you get this jig. Well, we actually glue them to something, play a movie past them on a nice LCD monitor, and then their forces can determine what the monitor gives. And we can tweak their gain and play all sorts of interesting games upon them. And then while we're doing that, record from the neuromuscular system. Okay? All right, so for today's talk, however, I want to focus dominantly on muscle. And I quote somebody who is one of my heroes in biology, and a few of you may know of him. His name is Bob Josephson. He's at University of California at Irvine, and he's one of these people who knows everything and then more. And he gave a talk recently, and he said, the most complex ganglia are inert. The most elaborate brains mute without the effectors to translate neuronal firing patterns into behavior. So when people think about the behavior of a robot or an animal or something, they tend to think about the brain as the sole part governing it. But because the brain is coupled to a neuronal system, which is coupled to a mechanical system, these should not be treated as separable. Okay? And I agree completely with Bob. And so I wanted to sort of focus on that issue in this particular case. And here is a picture of a hawk moth flying in mid-stroke in front of a flower. Now, if this, my real computer died yesterday. This is my older computer, so it may not show the movie appropriately. But this is an interesting preparation. I have a high-speed video camera filming this creature flying. To fly requires extremely high power output of muscles. So high that, in fact, in some flight situations, we see the evolution of a novel muscle type, which is capable of contracting many, 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 many times with a single neuronal spike. So it's a self-oscillating, extremely energy-demanding muscle type. Okay? These we have flying in front of a flower, and just to really tweak this animal, we're moving the flower at about 1 hertz while the animal's flying. So you'll see it at high-speed video. And what you won't see are the tiny wires that are doing recording in front of this. What is amazing is at 1 time per second, and this didn't show well, but let's try it one more time. At 1 time per second, that animal is tracking that flower phenomenally well, okay? Doing so while hovering. I challenge any of you to lick an ice cream cone moving back and forth your body length at 1 time per second, okay? What neuromuscular mechanisms permit this process, okay? So there are 2 approaches to understanding muscle design, and I'll give credit, importantly, to my postdoc, Michael Tu, who has black hair. It's important that I indicate this, who is my partner in this process. He is much more an experimentalist. I'm more a theoretician. So he says one approach, of course, is experimental. You take the muscles out. You do to muscles something to discover how they operate. And we're going to show you his data. And the other is, and you note the hair change here. This is his image of me, is you do modeling. And in fact, if you're doing it right, you're doing the 2 together. You're partnered in the laboratory, or you're working with somebody who's capable of building that bridge. So I'm going to talk about the 2 together. Okay, so let's see what these energy-demanding muscles are doing, and I need to educate you a little about how these animals work. Insect flight is controlled mostly by muscles that do not run the wings directly. Now, there are a few exceptions, but let me give you the sort of generic bug. The generic bug has a set of muscles that run in the fore-aft direction, the longitudinal direction in the thorax. And their job is to deform the entire thoracic skeleton on each wing stroke in such a way that when they squeeze longitudinally, the wings depress by a complex hinge mechanism. There's an alternate set of muscles that run vertically in the dorsoventral direction. They will counter-squeeze the thorax, and the wings will pop up. So it's sort of like this elastic oscillator that muscles are running, a deformation of the thorax. They're not tweaking the hinge of the wing directly, okay? Importantly, these muscles have two huge jobs to do. One is in each wing stroke, they have to create sufficient force to accelerate and drive the inertia of the wings downward. Shockingly, and I won't go into this, most of the force they need to expend is simply accelerating the wing, as if there's no air around them. Next highest force is the force required to deform the thorax. So if you cut the wing off, there's still most of the force they're expending is deforming the thorax. And next highest is the force required to produce aerodynamic forces. It doesn't seem like the best design, that the thing you're trying to do is create aerodynamic forces on a wing, yet most of your mechanical forces are associated with inertial dynamics, moving the wing up and down, and elastic mechanics. But what they're doing is setting up a mass elastic oscillator in the long run. So on the next stroke, that's very cheap, okay? So they accelerate the wing, deform the thorax, and they get elastic energy recovery on that backstroke. There are other very famous structures that require and rely on elastic interaction in cyclic muscle contraction. The most famous of these, of course, is cardiac muscle, which has a strong elastic component to it. Also periodically activated. Also expected to produce high power output for long sustained cyclic activity. So what Michael did is he devised a way to take an intact animal, and he actually decoupled the thorax from the rest of the wings and the skeleton, put a set of hooks in, and developed a force transducer method. It's actually a laser-based force transducer method that he can record the live forces while the animal is flying. And in fact, we go one step further, as you'll see later, is we'll actually turn the muscles on and off at will at any time we want. We can oscillate the thorax and do all sorts of combined activation and strain dynamics, okay? So we can record what they do, we can drive the muscles, and we can also record or measure forces. Record forces or drive motions, okay? Here is one of the results, and this is a plot of the relative force produced by these powerful flight muscles as a function of their relative length, okay? And the only thing I want to point out about this today is that, number one, this is actually an extraordinarily steep length tension graph by the standards of skeletal muscle. So skeletal muscle has, like Al Gordon and Huxley and Julian showed some 1966, a very nonlinear relationship between the length of the muscle and the force it generates. In skeletal muscle, that graph is fairly shallow compared to this. In cardiac muscle, it's quite steep. And in fact, if you overlay cardiac muscle on insect flight muscle, they look identical on the force length graph. Moreover, these animals are operating at a mean length and a range of lengths. So that's their mean length. They shorten and relax at a very steep part at the ascending portion of this length tension graph. And there's actually only one other muscle I know that has those characteristics, and that is, again, cardiac muscle. Operates on the ascending portion of the length tension plateau and does so with a steep length tension relationship, and does so periodically. Cyclically. Cardiac muscle does that in accordance with the Frank-Starling principle. That is, as your ventricles get fuller and fuller and fuller, right, the walls get ever thinner. But the force that you can generate by muscle declines as the cross-sectional area. And at the same time, the pressure or stress in the walls that you need to create that greater force is increasing as the ventricular radius of curvature increases. So the argument why cardiac muscle operates on that steep length tension plateau is in fact, a suggestion that it's an adaptation to the Frank-Starling principle. That is, as the ventricles get filled, you have ever more force to play with. As you ride up there, you have ever more force to play with on the next contraction. Okay? It might be the same for insect flight control. That is, they only sense their wing position at the peak of their wing stroke. So if you have a fixed temporal oscillator and you get a higher wing stroke amplitude due to a perturbation, you need more force to bring it back down to the same position and up again in the fixed amount of time. So another factor is that these animals operate on a very fixed pattern generator that runs the flight muscle. Almost always in our moths at 25 hertz, plus or minus very few hertz, 2 roughly. So a good way to regulate motion under fixed time constraints is to have more force if you have greater excursions, okay? Interesting problem. This is just a sort of standard measure of force behavior. That is an isometric measure. That is, the muscle actually is held at a length. We measure its capacity to generate force. We give it a new length. We do it again. But physiologically, nothing that I'm aware of is really isometric, fixed length, or for that matter, fixed force, isotonic, the two extrema of experimental methods we apply to muscle. So in situ, muscle is undergoing forces and patterns of motion that are varying in time, neither isometric nor isotonic. And Josephson developed an interesting way to sort of talk about such problems of muscles that are free to shorten and have forces change in time. And he developed something called the work loop method. Actually, it began earlier with Pringle and Machin in the 60s, also for insect flight. But it's been extended to many other muscle systems since then. Okay? And it works like this. Muscle is undergoing length change, right? So if I move my arm up and down, I'm undergoing length change, right? And I can have it go up, down, up, down, up, down. And in time, I may turn my muscle on here, here, here. So the motion I'm getting is like that. Now, in fact, if I take my arm, right, and I start to put external forces on it, I can drive it in one pattern of motion, and I can stimulate it at some other phase relative to that. So walking downstairs, right, my muscles are turned on but actively extended, right? Whereas walking upstairs, I may have muscles that turn on and are actively contracting. Okay? So it can go either way. And what you can do is you can measure the force at the same time that you're oscillating it and turning it on. So here it's going up and down and up and down. You turn it on, there's a big twitch, it turns off. There's a passive force, another twitch, a passive force, and I can play that game over and over again. So I might have a contraction followed by an extension. So you see the blue and the beige. And I can plot in that contraction the force versus length and compute the total work. So recall I go from long to short. So I'm going down this curve, and the integral of that is the work, the force length graph. And on the next phase, I can compute the energy I put into the muscle, right, to extend it. And the difference is the work that the muscle does. And multiplied by the frequency or divided by the cycle time, that gives you a measure of the power output. And in fact, you can go and measure the mechanical power output of muscles. And you can find that sometimes they are turning on in ways that they're not acting like we think muscle acts. Sometimes muscle acts not like a contractile element, but like a brake. So going down the stairs, muscle is a brake. You are turning it on, but it's actively extending. Okay? So you can either have network positive or network negative for muscle. Where do insects lie? Where does any system lie, for that matter, in the normal range of cycling lengths and the normal phases of activation? There is an underlying assumption that if you turn a muscle on, you may as well use all of the mechanical power output you can get out of it, right? So let's see what's going on here. We went and measured the in vivo strain pattern. It's close to sinusoidal, not perfect. We measured the phase of activation, and here's just a number. The frequency is 23 plus or minus 1.5 hertz. It's a very, very tightly focused pattern generator. Okay? The strain amplitude is that many fractions of the rest length. That should be 0.9 plus or minus 0.03. I just see a decimal error there. And the phase is about 0.48. Okay? The phase relative to the sinusoidal driving frequency. Okay? And here's what we find. This is a plot of the length of muscle, the phase of activation, and the mechanical power output is measured by that work loop technique. And this is actually an experimental plot. So we measured, we just controlled many different phases, many different lengths, and just did these motions, right? Different rest lengths, different strain amplitudes, and the like. And what you find is, lo and behold, there is this peak capacity to generate power output. So this is the surface that corresponds to this contour map here. And physiologically, you would hope that if you're going to turn the muscle on and run it at a length that maximizes the power output and at a phase that would maximize that power output. Right? That'd be great if you were using all of the power output of the muscle. But in fact, it doesn't operate that way. Physiologically, it operates at lengths that are below the peak length, as I showed you earlier, on the ascending portion of the length tension plateau. But I came up with some verbal argument why that makes sense. And they also operate at a phase, 0.48, that's way below the maximum realizable power of the muscle. So two things might go through your mind at this point. One is that insects are just primitive and stupid, and this is a bad design. I'll maintain they've had a lot more years of evolution than we have, okay? And been subject to natural selection for a considerably longer time. It might be, as one person suggested, this is Michael Dickinson at Berkeley, now at Caltech, suggested, well, it'd be stupid to run here, because if you're running here, you have no power reserves. The counter-argument is, so the smart thing to do is to run here, keep power reserves, and just turn it on in ways that you're burning energy. Because there's another underlying assumption is you turn a muscle on, if it's on for X seconds, you're just going to consume a certain amount of ATP. Because the ATP consumption rate is dependent not on whether you're shortening or lengthening, but whether you're on or not. Or so we think. And I'll add, or so we thought. Okay? So, why is it that they're running at this low phase? And in marches our conclusions right now. They operate at a phase and length that leads to rather low power output. Generally, and I wanted to point this out, these are running in a more primitive condition than another muscle type, the kind you find in flies, that has many contractions for a single activation. And we want to know, is there some characteristic of these muscles that is underlying this primitive oscillation capacity? Again, there's two approaches to understanding this. I showed you some experimental data. Now, it's gone against my hypotheses, basically. I thought, oh, they're going to run at a length that maximizes their force and at a phase that maximizes their power output, because I have an insect whose life is dependent on the mechanical performance of a massive muscle. It's about 30% of the body mass. It's the thorax. It's a huge muscle. Why not operate in a way that you use all of the energy capacity? So I wanted to approach this from a standpoint of more molecular motor modeling. It's that the assumption I said that is, it seems wasteful if, when you turn a muscle on, the amount of ATP you're going to use is independent on whether the muscle is going this way or that way. That is, I'm just going to cycle cross bridges according to cross-bridge rule. Okay? And cross bridges, as you know, are the molecular motors that drive muscle contraction. So let me delve down into a slightly different evolutionary problem, and that is the evolution of muscle modeling, not the evolution of muscle. The earliest model was Huxley's, basically, the two-state model. You have a molecular motor in the myosins that binds to some binding site, and on binding, there's energy is released, and this thing can undergo some contractile motion. That, in fact, like a lot of problems, 90% of our understanding derives from a very simple model. And I would maintain 90% of our understanding of sort of mechanochemistry of muscle is still contingent on the Huxleyan models. Okay? But we see the evolution of more complex ones from 3-state models, 4, 6, 14, 13, and even more. Each one of these models are delving into more and more conditions and states of the mechanochemistry of how myosin molecules interact with actin filaments in such a way as to account for a plethora of experimental data that's out there. Just like lots of other modeling efforts in biology, facts have a nasty habit of rearing their ugly head just when you trust your model most, okay? And just as my experience is, every time I give a talk, the facts appear in the audience and my models come crashing down. All of the models that I'm referring to on that first line fall into a category called mass action kinetic models. That is, what a myosin molecule on this part of the filament does is completely independent of what a myosin molecule is doing over another part of the filament lattice. Okay? In such a way that there cannot be any coupling. Thus, you could treat motor molecules as independent entities and accordingly develop very nice, straightforward differential equation models of the mechanochemistry of this process. That would be good only under one condition, and the condition is the following. If, when a motor molecule binds to the thin filament from the thick filament, upon that binding, it in no way affects the likelihood of anybody else's state transition, from unbound to bound or from bound to unbound, either way. You can only do that as long as you're assuming independence. Unfortunately, you can only assume independence if when one molecule binds, it doesn't distort the filament lattice in any appreciable manner. Sadly, in 95, 96, and 98, a set of papers came out. One was by Yale Goldman called Filament Compliance, Are You Pulling My Chain? It was one of the better titles I've seen in a long time, which was a question saying, if you're pulling my chain of binding sites, that means that this sort of little panic kind of settled into the modeling world, which was, if my binding sites are moving when a motor molecule binds, it means I can't assume they're independent. If I can't assume they're independent, my mass action models have a little bit of a challenge to deal with. So the question is this a problem? Number one. Number two is, can we develop models to account for this and to account for the possibility that the state transitions are not independent of the fact whether you're elongating or contracting while you're on. Filament compliance gives us an inroad to possibly understanding this peculiar phase issue I raised earlier. So I'm going to talk about a different class of models. They're Monte Carlo models. Some are Monte Carlo Markov chain models. There's a variety of these. A Monte Carlo model is a lovely and very French way of saying brute force simulation. It takes very simple building blocks and rules for how motors behave. And so what I'd like to do is to outline this for you. Again, I probably should have done this in a slightly different order, but let me remind you of the geometry of muscle and our thinking about it. Here is a light micrograph of muscle, and you can see, of course, a regular banding pattern here corresponding to repeating sarcomeres. The sarcomere is the functional unit of a muscle, subtended by a Z-line or Z-disc, with thick and thin filaments interdigitating. Okay, thick filaments containing the motor molecule, the motor molecules with extending off the rod. So here you have a Z-disc, thin filaments interdigitating with thick filaments. Many of people in this department certainly know this inside and out. And so you have this interdigitating array of filaments, and radiating outward from the thick filament core are these motor molecules, myosins, and they have a helical packing structure. And they face a set of these actin filaments. They're actually a twisted chain of F-actins here. Okay? And our image of this is you turn a muscle on, and all these motor molecules to sort of reach out, bind onto the thin filament lattice, generate their force, and let go. And so our notion of this is that they basically are running at high duty cycle. That is, most of the motor molecules are binding. I mean, it would be profoundly stupid, you know, if you didn't use most of your motor molecules, right? I mean, you've got them all there. I mean, heaven forbid you should use about 80% of them, right? 50? Anybody have how many say it's over 50%? How many say it's under 50%? We have a bad case of undervoting here. How many are undecided? It was really undervoting. Okay, so there's the majority have no vote. Okay, which is fine. Let me explain something to you. The filament lattice our notion is there's lots of binding with all the motion in the motors, as opposed to very little binding with lots of motion in the filament lattice. Okay. So if the filaments are not deformable, there's no interaction between cross bridges, and mass action models are appropriate. However, with the filament deformations, we do have a problem. So let me look at a Monte Carlo simulation and talk about this of staying attached. So let's look at the interaction of this guy with this filament lattice here. Okay. And what I do in a Monte Carlo simulation is I throw a pair of dice, or a dice, right? And that has some probability distribution. And I'm a MATLAB fiend, so here's my code if you care. But if I want to ask what is the likelihood of binding to that, I have a transition rate that is contingent on some energy conditions of that molecule. You have an on rate that may depend as well on the time steps. So if I take very tiny time steps, I need, it's very low probability that I'll undergo a transition. If I take very huge time steps, probability is going to be 1 that I undergo that transition. And it will depend on where that molecule is relative to its binding site. So if the molecule it's got its nose right on the binding site, there's a very high probability that it's going to find it under thermal forcing. If the binding site is between my nose and that wall, and I have an extensibility of 2 nanometers, the probability is not 0, but for all practical purposes it is, right? So the likelihood of binding depends on the time step I'm going to choose, but also on the spatial position of that molecule, okay? So if a molecule does bind, what forces can it generate, and how does that affect the spatial positions of all other binding sites? So what we do is we set up a little system of motor molecules, okay, each connected by a spring whose stiffness is in fact reflecting that measured experimentally by Wakabayashi, Goldman, Eisenberg, and others, okay? And they can attach to putative binding sites. In the case of this, we actually have experimental estimates. This is now probably much higher than I have in this slide here. It's probably closer to 10 piconewtons per nanometer. But let me just go ahead with this number at this point. And we can do a balance of forces about all nodes. And so all rightward stretches of springs are balancing all leftward stretches of springs. I throw inertia to the gods of the big world, leaving it out of my domain here. This is a tiny, tiny world, the level molecules, okay? And in fact, we then have rules for cycling. We need then rules for cycling cross bridges. It's an interesting fact that these cross bridges extending off of one side of a thick filament are spaced every 47 nanometers, 43 nanometers, okay? And the binding sites are every 37 nanometers, okay? That's an interesting observation in that the fraction of cross bridges that are within, say, 2 nanometers of a binding site is about 15%. So because of this misalignment, so to speak, of cross bridges with binding sites, very few are within 2 nanometers. It turns out for our best estimates of the likelihood of binding, you need to be within, in this kind of myosin, I should say, within 2 nanometers or so of a binding site to get over that sort of 20% likelihood of binding, to be in the top 80th percentile, right? So in fact, very few could be bound at any one time. And that seems to be what's emerging in the literature today. Certainly what Joe Howard was suggesting, I think Mike could probably correct me, but it's in that range. There are other myosins, I should suggest, that can reach 30 nanometers or so, some people suggest. So here we have a Monte Carlo model we're going to do, we need to get the state transitions clear. Let's set up the geometry appropriately with 43 nanometer and 37 nanometer binding sites. There's that issue of the vernier, and I said this is really old now. We're way back in 98. It's the previous century. I feel ancient at this stage, okay? This is done in collaboration with Bryant Chase, okay, who's now at Florida. And we ask how these cycle and how the forces are distributed. So the idea, and we're going to use a very simple model, there's a four-state model we're working on now, is that you have ATP hydrolyzed and a cross bridge is preset to bind. And we'll call this state 1. It's unbound. It's snooping around with the help of thermal fluctuations for a binding site. So the kind graces of temperature and water molecules are providing the motive force for bombing this guy around to find a binding site. It may bind with some probability that is dependent on how far that binding site was and how stiff that motor molecule is. So if the motor molecule is made of titanium, you might need to be within a few angstroms of a binding site. If the motor molecule was made of mucus, you could have some considerable distance, but it might not be very effective. It binds, and on binding, the phosphate falls off. You generate a power stroke. There's a conformational change in the molecule, either a rotation, I think it's the dominant view. In some motor molecules, there's a shrinking, as suggested by a recent job candidate in physiology and biophysics, Mike Rock, I think. Really interesting data. It unbinds. Rehydrolyzes ATP, uses that energy of hydrolysis to reset the engine, does this process again, and continues this ad nauseum. The state transition model is you go from unbound to bound with some rate transition, drop the phosphate off, you're in the strongly bound mode, and then you hydrolyze ATP, this enzyme, myosin ATPase, hydrolyzes ATP for subsequent strokes. So you're generating force at the expense of energy, the sneaky trick of myosin ATPase molecules, and probably G proteins, as it turned out, is that they hydrolyze ATP, and that hydrolysis energy is manifest as a mechanical distortion. And it's that mechanical distortion which provides the motive force. Hence, it's a chemomechanical process, taking chemical energy of hydrolysis to perform distortional work in a molecule. The forward transition rates, let's just talk about those, depend on your distance from a binding site, your distortion of a cross bridge, and the reverse transition rates are calculated by standard equilibrium thermodynamic calculations. That is your delta G associated with these states, given no mechanics of the molecules. We use a Monte Carlo simulation with stochastic forcing, use those cross-bridge rules, do that force balance on the number of motor molecules we're using. We use 20 collinearly in this case, and turn the crank, holding the ends in any way we want, controlling calcium levels in any way we want, and calculate average forces in instantaneous motions. And what we find is if you plot, and this is using modeling not as a predictive tool, but as an exploratory tool, right? So if you plot, you're going to explore what are the consequences of filament compliance, cross-bridge spring constant to the capacity to generate tension. What you'll find is, in fact, that they generate force at high force at intermediate stiffnesses of the cross bridges and the filaments. And in fact, if you look physiologically, we're right about there for what we think cross-bridge stiffness and filament compliance is like, right where the peak tension is. This region of high force under low compliance is done because the molecule is so compliant you can recruit lots and lots of cross bridges, right? You're just dragging all the binding sites around in such a way to get lots of alignment and lots of favorable alignment in some cases. The problem is you need lots of energy to do that. You're cycling your cross bridges massively as your filament is realigning all the time. And so if we divide it by the amount of ATP we calculate is used, the number of state transitions we have to go through to get there in a fixed amount of time, in fact, that's a very poor domain to live in, right? So you're living right about there. And in fact, the newer estimates of force generation, or spring constants of myosin, skeletal myosin, is closer up to here now. When we did the simulations, it was about maybe a tenth of what we think it is today. I'm going to skip this, but the temporal dynamics matter a lot. Okay, here's the trick. Binding rearrangement happens in response to cross-bridge forces. The geometry and mechanics can be tuned to maximize force as one measure of efficiency, or force per ATP molecule. There's some issues, however, with what we did. That was old. That was 98. We were naive. Okay, so you can only say this when you're the author. Daniel was exceedingly naive. He should have realized the force balance he developed was likely wrong. I had one thick filament, one thin filament. Yes, I had the spacing right, but it was a 1-to-1 ratio. Okay? Filament ratio does not represent the true structural values in three-dimensional lattices. Okay? So Daniel blew it there. Model had a few interactions. It was 4. Because I had such a low binding likelihood, and I only had 20 cross bridges collinearly for a half sarcomere. I could have made a longer one, but it would have been physiologically stupid. I only had 4 out of 20, roughly, molecules interacting. So I had to do lots and lots of stochastic simulations to get some averages. And any temporal patterns were likely hidden in the behavior of the stochastic mess I had generated. I'm really trashing this guy. With such few interactions, one can't realistically add damping, mass, other factors to the system because there's no way to easily scale this problem. We need more bridges. We need better packing. So, with an undergraduate in applied math, actually, his name was Mike McPherson, we derived the model for a three-dimensional lattice structure of interacting motor molecules. Michael went on, by the way, to get the President's Medalist for UW in 2001, and he's now at Stanford abusing other mathematical biologists there. So what we're going to look at is the tessellation of cross bridges, and looking end on now, cross-section through a muscle. I apologize for the sort of schematic view. Each one of these is a thick filament with radiating cross bridges. You're not seeing the three-dimensional spiral structure. But we have that implicit in the model. They face thin filament here, thin filament at each of these vertices, okay? Well, this cross bridge here, if it generates force on this thin filament, could interact with that thick filament there. So the tessellation is a bit of an issue. And I realize I'm going to go really fast now. So I'm going to take a subset of this and do a mirroring where I interact cross bridges of filament A, right? Coming off the thick filament A, it will interact with this thin filament B, but I'll mirror it on this filament here. And so I'll be able to sort of tessellate this set of hexagons in a way that I balance the forces in as close to a 2-to-1 ratio as I can get. So we use 13 actin filaments, 90 nodes each, 30 myosin filaments with about 1,300 some odd nodes, say 1,400 nodes. And we're getting close to the packing that's present. Okay. And let me just give you the results now. What we find is, in fact, this is now an actual simulation of the results with the virtual myosins turned on, we say. That is, the mirrored edges of this tessellated set of hexagons, okay? And it turns out it doesn't matter a lot, but it matters somewhat in the early transients of force generation that you have that appropriate force balance, okay? So temporal dynamics matter. But what the key thing is we set something up with a lot more cross bridges. Now I want to come back full circle in the remaining minute and say, why did I get that phase? This is where it all began. And I didn't want to lose track of the fact that I was measuring a muscle at one point and I had a question. And the question is, why operate at a phase that is suboptimal? I couldn't use my little degenerate two-filament model to oscillate it and query the ATP because the stochastic noise was so large it was impossible to pull the signal out. So we grew the model as best we could, redid this in the following way. We're going to ask a question. I will turn calcium on for some amount of time on my virtual muscle. Now I've got a set of filaments interacting. And then I will turn it off. And I'm going to do it at exactly the cycle rate and the mechanics we know to exist in our moth muscles. Okay? So it's about 25 hertz, which is 40 milliseconds. I'm going to oscillate the muscle. I can control the end conditions because I made a spatially explicit model. Okay? So I'm going to oscillate it. I'm going to just turn calcium on and off, and I'm going to adjust the phase of my motion relative to my calcium signal. This isn't the best calcium dynamics, but it's going to answer a certain question. And my question is, how does the force, power, and energy depend, from our modeling standpoint, on the phase of calcium activation? Okay? So here we have a plot of the relative ATPase rate, that is the number of ATP molecules consumed, right, per cycle. And what you'll find is it is phase dependent. And the reason it's phase dependent is your transition likelihoods depend on whether you're extending the spacing between binding sites because of compliance or condensing the spacing between binding sites because of compliance. So in fact, your transition likelihoods are dependent on whether you're going out or in. That would not necessarily have been the case in a rigid model, a model of rigid filaments. We find that the power is peaked at about 0.3 something, which is almost exactly what we measure to be the experimental maximum power. But that's not where they were operating physiologically. So what we then said is the physiological phase of activation was 0.48. What is the ratio of the mechanical power output you get relative to the chemical power you're providing? Maybe that's a more interesting question. So we literally took the ratio of the mechanical power to the ATPase predicted by the model. There's the phase for measured maximum power output that we had, right? And then what is the power per ATP? And here we find, in fact, it goes very negative because you have negative work done here. But in fact, they're operating basically at the highest possible efficiency during normal flight. The flight control of these moths is modulated not by their frequency, but by the phase of activation relative to the lengthening and shortening of these muscles. And in fact, they do have power reserves, but they're probably running at a way, possibly I should say, running at a way that is using the least amount of energy per power you can produce. I want to conclude by saying this is actually not to be believed. And the reason you shouldn't believe it is I need to measure experimentally the ATPase rate of live muscle as I adjust the phase of activation. We actually have an experiment planned, but nobody in the lab right now wants to do muscle mechanics. They're all on the neural side. So all the equipment's there, the question's there, and I'll leave off the oscillations. Let me just go to the conclusions and say thanks. Spatially explicit models are easy to implement. They account for movement of binding sites in response to cross-bridge forces, reveal mechanical tuning for force and timing. They scaled upwards to examine force balances on larger systems and provide a hypothesis for variation in ATPase rate with phase of activation. These models operate far from a peak, but may not be bad designs, may in fact be very good designs. And I didn't show you that we are getting oscillations if we tune the kinetics just right. And such models owe their deep and profound thanks to the National Science Foundation, National Institutes of Health, MacArthur Foundation, and the Joan and Richard Komen Professorship. Thanks.
Thank you.